Hello, class. Welcome to the next unit. This is our last unit of the class. We'll have three more chapters that we're going to go over, and then we are officially done. So, let me get the slide up, and we will get started. Okay, for today, we are going to go over Patterns of Inheritance, Chapter 10. So, in the last unit, we finished off with meiosis. Meiosis is the process of producing gametes, haploid cells, or sex cells. These are cells that are used for sexual reproduction. An egg and a sperm come together to produce genetic variability and produce an offspring. Now, with that knowledge of meiosis and how it works and how it produces genetic variability, we're going to talk about inheritance. So, genetics can explain and predict inheritance patterns. A gene is a segment of DNA, like we've been talking about for the last couple weeks, that carries the instructions for synthesizing a protein. Genetics can explain why these people look different. Analyzing their genes can also predict the appearance of their offspring. So genes are important. Genes can help us determine why individuals look different. Sometimes this is just a simple one or two genes. Sometimes it's complex, and we'll go over that later today. But genes can help predict inheritance. It can help predict what what's going to be passed on to the offspring. Genes also predict body function and health. Most genes include proteins that have nothing to do with the outward appearance. The enzymes essential to our lives are also the products of genetics. Studying genetics allows scientists to breed superior crops and doctors to track genetic illness. So, not only can genes be used to predict the appearance of offspring, but they can also be used to one, produce superior crops. You might have heard in terms of food, genetically modified organisms such as fruit. So we can use genes, use inheritance loss to make genetically modified fruit. And genetics also can be used to track genetic illnesses. So genetics has a wide variety, and it's because one, they encode for special proteins, all the proteins in our body. And with that, we can use it to check our health, check our appearance, and see what could be passed through to our offspring. So, chromosomes are packets of genetic information. Recall that chromosomes is a piece of DNA containing many different genes. Each gene locus, I'll get my highlighter out. Each gene locus is its location on the chromosome. So whenever you hear about a locus, oh, wrong thing, it's the location of a gene on a chromosome. Right. When two haploid sex cells fuse, a diploid zygote with two full sets of chromosomes, it's formed. So remember, we have two in a diploid cell, we have two full sets of chromosomes. So in this picture right here, we have gene A and gene A, gene B and gene B. And this is when the DNA is already duplicated, already replicated. So we have two full sets of DNA. Chromosomes are paired in diploid cells. Each chromosome is a member of a homologous pair. Humans have 22 homologous pairs of autosomes and one pair of sex chromosomes. Remember, our sex chromosomes, these are X and Y. In males, that's X, Y. And females, that's X, X. All the other chromosomes are going to be what's autosomes. So we have 22, it's really 23 pair of chromosomes altogether. Each chromosome has the same set of genes, but could have variations in the gene. That's what we're going to go over next. Diploid cells have two alleles for each gene. A member of a homologous pair have the same genes but might have different variations, different versions, called alleles of those genes. So an allele is a different version of a gene. Keep in mind, a different version means that it still encodes for the same protein, but it could have a slight different structure in that protein. Same proteins being made, but it could look a little different based off of the allele. And we're going to see examples of this later. These alleles might be identical, gene A, or different, gene B. So again, in our example here, we have gene locus A. Within the moth, with the pink chromosome, we have a big A, and in the blue chromosome, we have a big A. So those two are the same. But in gene B, notice how the pink chromosome has a big B, and the blue chromosome has a little b. The big B and the little b code for the same protein, but a slight variation of that protein. So alleles are just different versions of the same gene. Since we all are diploid organisms, we have two different alleles. They may be the same exact gene, they may be slightly different versions of the gene. But because we are diploid, we have two sets of genes that could be different. Now, as we go into inheritance, it's important to talk about a science that's called Mendel. Mendel uncovered the basic laws of inheritance. He used pea plants to study heredity. What he did is he took pea plants and he hand pollinated them, allowing him to control the plant breeding experience. So he took pea plants, he wrote down their characteristics, such as how tall they were, the color of the seeds they produced, and so forth, and would self-pollinate them himself to see how does inheritance work. Now, the offspring gave Mendel clues about the genes of the parents. So we're going to see an example right here. Let me get my pen. So this these are the parents. We have a green seed and two yellows. What's happening here is Mendel self-fertilizing. So he's taking the pollen of the plant and using it to fertilize its own flowers. Keep in mind, plants can do that. Plants can do produce sexually, but they can self-pollinate. They can pollinate their own flowers and produce identical copies. Or produce near identical copies of themselves. There's still some genetic variation since it's still sexual reproduction. So in each of the examples, Mendel self-pollinated the plants. In the green plant here, when it was self-pollinated, all he got were green seeds. The same happened in this first yellow seed. There's self-pollination, all he got was yellow. So these are examples of true breeding plants. Self-fertilization yields offspring with the same seed color as the plant. True breeding plants, down here, plants that produce offspring identical to themselves. So a true breeding plant produces offspring identical to themselves. And the last example, I'm gonna get my eraser real quick. Erase those. And there's the last example here. We take that yellow seed, self-fertilize, but we get two different seed colors. We get one green. So notice how in this we have one green seed here. This tells us that this is a hybrid. Self-fertilization mixes the seed colors. Hybrid plants outwardly resemble true breeders, but produce mixed offspring. Say true breeding plants will always produce identical copies of themselves, but a hybrid plant may not. They look like they're true breeding plants, but they can produce mixed offspring. We'll talk about why that is in a second. So before we do that, let's talk about different types of alleles. We have what's called dominant alleles. And for this, I do want you all to know the next couple of terms. Dominant alleles exert their effects whenever they are present. Crossing a yellow seed with a green seed always yields some yellow seeds. So for any time these yellow seeds are used, there's always going to be some yellow seeds produced. So we can see if we split this in half, in our first example here, yellow seed is used, and we get all yellow. In the second example here, again, yellow seed is used, we still get a little bit of yellow. Yellow seed color is therefore dominant. If you always get that trait, that means that allele is dominant. If it's always present in the offspring, it's dominant, as long as the allele is present. Next are recessive genes. A recessive allele, like um, like green seed color, is one whose effect is max if a dominant allele is present. So again, split this in half. Oh, let me redo that. Let me get my eraser real quick. There we go. Use the blue and example one. We have the green seed here, but notice all yellow. So now we know this green color seed is a recessive trait because even though it's present, since we have yellow, we get yellow seeds only. Even if for this one we have green seed present in the second example, but we get some yellow still. So a recessive gene, a recessive allele, its effects will be masked by the dominant. The dominant always takes over. The only time you will see a true recessive um, trait is when both the offspring has both recessive alleles. We'll see that in a second. So, why do some plants produce both yellow and green seeds? So this is what I kind of hinted at. The answer has to do with each plant having two alleles for each gene because they're homologous pairs of chromosomes. Remember, a diploid cell has homologous pairs of chromosomes. Each chromosome has the same gene, but they're going to have different alleles for that gene. And that's what's going to help us determine why sometimes the seeds produce both yellow and green seeds. So now we have two terms, and I do want you to know these two terms: genotype and phenotype. The genotype represents the individual's two alleles. The genotype represents the individual's two alleles. Oh, no, no. Come on. There we go. The genotype confers or produces the phenotype, which is the physical appearance. For example, in observable characteristics such as seed color. So with this, I want to write here. Actually, let me do this in block. I'm gonna write up here, genotype, think the genes, slash alias. Well, I spelled that very wrong. Come on. I don't know why. Give me 1 second now. So genotype produces the genes or the alleles, while the phenotype produces the physical characteristics. My app is not working. Let me try to get out. So knowing that, make sure you know what the genotype and phenotype is. Let me restart the app real quick. With that knowledge, we're gonna start looking into um, why sometimes you get mixed offspring. Let me get back to where we were. There we go. So again, if I zoom in, let me write that down again. Genotype, you go to genes or the alleles. Phenotype, it goes the physical characteristics. And that's what I want to know. This is how you determine each one. G for gene, P for physical. And we'll highlight that too. I forgot the E in this word, and that's okay. So with this, we also have other terms that I do want you to know. This will be on the test as well. Homozygous dominant, heterozygous, and homozygous recessive. So homo means same, hetero means bien, homo means same. So in a yellow seed, we could have a homozygous dominant. Homo means same, dominant has the dominant allele. That means it has two dominant alleles, which is in this example, two big Ys. Whenever we talk about genes and alleles, a lot of times you're going to see these letters. And a lot of times you see an uppercase and a lowercase, a big or little. Dominant genes or dominant alleles will most of the time be the capital or the big letter. So in this case, the big Y. The recessive genes are going to be the small letters or the lowercase letters. In this case, the lowercase y. So in homozygous dominant, the genotype, so this is genotype, this is the phenotype. And it says it on the slide too. Realize. Let me get my eraser. I'm gonna erase that. So we have the genotype here and the phenotype here. So the first one is a homozygous dominant. We have two big Ys. It produces a yellow seed. Homozygous, so it has the same two alleles. Then we have heterozygous. That means we have two different alleles. We still have the big allele, the dominant allele. That's where we get the dominant phenotype, which is yellow. But we also have the recessive allele too. And lastly, we have the homozygous recessive. Homozygous recessive has two recessive alleles. Because it has two recessive alleles and no dominant, we finally get the recessive trait, which is green. So homozygous means same, heterozygous means different. Homozygous dominant means two dominant alleles. Homozygous recessive means two recessive alleles. And if you're homozygous recessive, that means you're going to show the recessive trait, in this case, green. Now, look at the offspring to determine the genotype of the parent. Individuals with the same phenotype can have different genotypes. So in this example here, I'm going to split this here. We have example 1 on this side, 2 on this side. If we cross a green and yellow seed here, we know the yellow one is going to have little y, because in order to show that green, you need a homozygous recessive. It can't have any dominant allele. Excuse me. And the yellow one, we know we have one big Y, but we don't know the other one, because remember, we only need at least one to show. When these two plants are reproduced, when one pollinates another, we get all yellow seeds. So that's telling us something. In the next example, we have green, so little y, and then we have yellow, big Y, and something else. But this time, we get some green and some yellow. So that's telling us like, hey, something's going on. Somehow the offspring are getting the recessive trait. How does seed color genotypes work? The seed color gene encodes for a pigment metabolizing enzyme. The dominant allele, which is the big Y, is active and produces yellow color. The recessive allele, the little y, is inactive. In the absence of the enzyme activity, the seed stays green. So in this first example here, we have a little y. We don't have any of that enzyme being produced, so we only get green. But in the other two here, we have big Y something, big Y something. Since it has at least one dominant allele, we get that working enzyme that produces the yellow color, so we get the yellow phenotype. Now, take a second to distinguish between dominant and recessive, heterozygous and homozygous, phenotype and genotype. Okay, let's go over this. So dominant alleles always show their characteristic as long as they are present. Recessive alleles only show their characteristic when the dominant allele is not present. They have to be by themselves. Heterozygous means you have two different alleles. Homozygous means you have the same type of alleles. Big Y, or little y. Phenotype is the physical characteristic. And lastly, genotype, or for phenotype, physical characteristic. So it's seed color that can be red, sorry, yellow or green. And genotype is the actual genes. So they can be big Y for homozygous dominant, big Y for homozygous, or for heterozygous, excuse me, or little y for homozygous recessive. Now, with all that said, how does this go into predicting inheritance? We have what's called a Punnett square for this. So Punnett square uses the genotypes of parents to reveal what the offspring may inherit. May inherit. It reveals what the alleles the offspring may inherit. So in this example, we have the female here and the male here. This female has the genotype big Y, little y. So remember, it's a diploid cell, so it has two copies of the same gene. During meiosis, though, those genes are split. So we have one big Y here and one little y here. And the same happens in the male. We have big Y, little y. So we get one big Y here and one little y here. This right here is our Punnett square. It's gonna have a 2 by 2 little square. So on the outside, on here and here, we put the different alleles each parent is offering. So mom gives you a big, in this example, is given a big Y and a little y, and dad is doing the same, given a big Y, little y. Punnett squares show the genotypes. In the example, a female heterozygous, which is a big Y, little y, for seed color, is crossed with a male parent, also heterozygous, big Y, little y. This is called a monohybrid cross, since both parents are heterozygous. I do want you to know what a monohybrid cross is. Monohybrid cross is when you cross heterozygous parents. Both parents have mixed alleles. Punnett squares show how alleles separate during meiosis, and that's kind of what I got to earlier. When germ cells divide by meiosis, the gametes receive one allele per gene. So that's why, oops, too far. With this example, we get one allele per gamete. For seed color, there's an equal chance of receiving either a big Y or a little y from both parents. Punnett squares show how alleles get back together after fertilization. A gamete from a female parent, excuse me, and a gamete from a male parent then unite in fertilization. So this box here is showing them reuniting. Each option here gives you an option that could happen in the offspring. A big Y goes down here, big Y goes here, so you get big Y, big Y, big Y. But we could also have the big Y coming all the way down here with the little y coming here, so we get big Y, little y. Excuse me. Offspring could inherit two dominant alleles. And now we're kind of breaking it down. So we see in the example right here, we have the big Y from the mom going down to the first box, and the big Y from the dad also going to that first box. So one genotype that can be produced is big Y and little y. Big Y and little y. So this offspring will be homozygous because it has the same alleles, and dominant because they're the dominant alleles. So it's homozygous dominant. Offspring could inherit a dominant and recessive allele. In this genetic cross, one gamete could carry the dominant allele and the other could carry the recessive. And notice here we have two different ones. In this first one, we get the recessive from the mom and the dominant from the dad. But in this one down here, we get the dominant from the mom and recessive from the dad. both both have different alleles of dominant and recessive, thus it's heterozygotes. So we can get two different ways to get heterozygotes in this monohybrid cross. Offspring could inherit two alleles, two recessive alleles, excuse me. In the genetic cross, both gametes could carry recessive alleles. So in this bottom one, the only green seed, you can get a green seed because the mom can give a recessive allele and the dad can give a recessive allele. Anyway, for the recessive trait to be present, or not present, but for the recessive trait to actually show, there can be no dominant alleles. So in this case, it could happen. You can get the recessive trait, the little y from mom, and the recessive trait, the little y from dad, causing that seed to be green, causing it to have the recessive phenotype. Punnett squares show the expected proportions of offspring. This Punnett square is a prediction that shows the relative proportion of offspring, phenotypes, and genotypes. So here we're going to look at two things. So give me a red. We have the genotype ratio here. I'm going to zoom in too. Where based off the Punnett square, we get only one YY, one YY, one dominant big Y, two big Y little y's, two big Y little y's, and one little y, one little y. So that's how we can do the genotype ratios. Doing homozygous dominant, we get one. Heterozygous, we get two. And homozygous recessive, we get one. So it's a 2 to 1 to 2. Sorry, a 1 to 2 to 1 ratio. But on the flip side of that, if we erase all of this, apparently this thing does not like the eraser. Let me get out again real quick. I'm going to try this again real quick. So with the Punnett square, we can actually show the expected ratio, the expected outcome of the cross. And the phenotype one, notice how we get 3 yellow, so 3 yellow and only 1 green. So it'd be a 3 to 1 ratio. So with the Punnett square, I do want y'all to be able to label what's the genotype ratio and what's the phenotype ratio. If your genotype is homozygous dominant, heterozygous, homozygous recessive. And phenotype are just the two different ones, two different phenotypes. Punnett squares can help us answer Mendel's question. If a seed color is dominant, why are some seeds green when a yellow seed plant is crossed with a green seed? So in this example here, let me get my blue out. In the first example, we do all yellow seeds. It's all yellow seeds. When we have alleles, we know the green is little y, but the yellow is big Y something. If there was a little y, so in the second one, let me go over that. Little y, big Y, little, excuse me, went too far. Let's say in this case, we have the yellow seeds has a recessive allele. Because it has a recessive allele, it can actually produce the green seeds as well. But if the first example has both dominant alleles, it's homozygous dominant. It cannot pass the recessive allele, the green trait. Thus, all seeds have to be yellow. So when you do what's called a test cross, you're trying to see what genotype the yellow seed is. If it produces all dominant offspring, or offspring with all dominant traits, it's a homozygous dominant because it cannot pass recessive traits. But in example 2 here, if you get offspring that do have the recessive trait, that means the yellow seed has to have a little y. And I'll show y'all an example here too, without erasing. So I'm gonna do 1 up here, 2 down here. To kind of get you thinking about this in the right way, I'm going to draw my Punnett square. So we know the dad is a little y, little y. We know we have big Y up here and something. So if we bring down that big Y, we have big Y here, big Y here. And in the dad, we have little y, something with a little y, something with little y. Now, if we put a big Y here, we get big Y, big Y. So for the genotypes, for big Y, we get 0. There's no homozygous dominant in this cross. We do not produce any big Ys. Remember, we're looking at the products in each square. For the numbers of big Y and little y, we have 1, 2, 3, and 4. All 4 offspring have to have a big Y, little y. And we look at homozygous recessive, we have 0 again. So that's our genotype ratio. No homozygous dominant, all heterozygotes, no homozygous recessive. But in phenotype, every single one has a big Y. That means for yellow seeds, all 4 are yellow. But for green, we get none. So if the mom has both, or the yellow seed has both dominant alleles, it produces all yellow seeds, which is why down here we get all yellow seeds. There's no chance of getting the offspring, no chance of getting the green allele. Then the other example, let me draw my Punnett square again. Draw a little bigger this time. You know, one has 2 little y's, and the big one we're going to say has big Y and a little y. Again, bringing down the top ones, big Y here, big Y here, little y here, little y here. And then going across, we have little y, little y. On the bottom, we have little y, little y. So for homozygous dominant, we get 0. For heterozygotes, we get 1, 2. So we get 2. For homozygous recessive, we actually do get 2 in this case. So we have a 0 to 2 ratio. For the phenotype for yellow seeds, remember, yellow is going to produce anytime there's a big Y. We have 2 big Ys, 2 with big Y. So we get 2 yellow seeds. And green is when it's homozygous recessive. And we do have homozygous recessive. We have 2. So the 2 to 1. So in this second cross, in order to get some green, you do need that recessive allele in that yellow plant. So that is why using Punnett squares, we can determine what the genotype is of the parent, knowing the offspring. We can kind of do the reverse. We can put in, we can go like, oh, if there's any type of recessive phenotype, you have to have recessive alleles in both parents. Offspring can reveal the parents' parental genotypes. So homozygous dominant. This I went a little too far, so I'm going to skim through this part. And this goes over the same thing I just did. If a cross between a yellow seed plant, big Y, or a little y, big Y, little y, excuse me, and a green seed plant, little y yields all yellow seeds, the yellow seed parent is homozygous dominant. So in the example, if all yellow seeds are produced, you have a homozygous dominant plant. And that's shown in this example here. We only have big Y, big Y. Big Y is the only thing that can be passed down by that female. So you get all yellow seeds. No chance of green seeds because you don't have the recessive allele from the mom, from the yellow seed. Offspring can reveal the parent's genotype for heterozygotes too. If a cross between a yellow seed plant, big Y, or big Y, little y, and a green seed plant, little y, yields some green seeds. Some green seeds, the yellow plant is heterozygous. And that's because in order to produce these yellow seeds here, you have to have a recessive gene. So that yellow seed must be a heterozygote. It must be a big Y and a little y to be able to pass down that little y and thus show the recessive trait. In this case, the green seed. Mendel's laws can be applied to human traits as well. Punnett squares are also useful for tracking inheritance of genetic disorders such as cystic fibrosis. So in this case, we're making the assumption that cystic fibrosis has a dominant allele and a recessive allele, with the recessive being the gene for the disease, for cystic fibrosis. So we can do the same thing. If you're going to hear this word a carrier means that the person has one gene with the disease, but one gene without, but they are not expressing it. Since they have the dominant allele that's healthy, they are a healthy individual. So in this case, we have two heterozygous parents, big F, big F, little f. They're both carriers, so they both have the recessive gene for cystic fibrosis. So here we get FF, healthy non-carrier, 2F, big F, little f, which are carriers since they have the recessive trait. We get one individual with that recessive phenotype, which is cystic fibrosis. So Punnett squares and Mendel's laws can be used for human traits. As we'll see later on, though, this can get pretty tricky, though. Not all genes or not all traits or diseases are determined by simply one gene. So here's a question, and I do want you to try to answer this. Cystic fibrosis is caused by a recessive allele. The healthy carrier, remember, carrier is somebody with a dominant and a recessive allele. They're heterozygotes. So a healthy carrier and an affected individual have a child. What is the chances of the child being affected? Pause, think about this. We'll get back to it. Oh yeah, with this, we have a healthy carrier. Maybe a healthy carrier means that they're heterozygous. They have one healthy allele and one recessive. And an affected individual, for this case, since it is a recessive allele, in order to them to be affected, they have to have both recessive genes, both are recessive alleles. So they're homozygous recessive. We do our Punnett square, do a cross. For the first one, we do big F and little f. Remember, we're separating them. Little f and little f. We start with a big F, we bring it down to big F, and then we start with the little f on the right column. Little f, little f. And then on the top row, we have little f going across, and the bottom we have little f, little f. So we're asking what's the chance of the child being affected? To be affected, it needs to be a homozygous recessive since we're dealing with recessive alleles. Remember, the only time a recessive allele is shown is when there's no dominant allele. So in this case, we have two. 2 out of 4 of the offspring can be affected. 2 out of 4 means we have 1/2. So you have 50% chance of this individual of the offspring having cystic fibrosis. Now, now that we know Punnett squares, now that we know how to use them, the difference between dominant and recessive genes, let's talk about dihybrid crosses. Dihybrid crosses track the inheritance of two genes at once. Two genes on different chromosomes can be combined into one Punnett square. So all we did in the beginning is we just looked at one trait at a time. But now you'll notice we're going to have two different genes we're looking at for all these individuals, with big Y and little y, or the Y's being for color, but we also have R now. R is for the shape. Big R is round, that's the dominant allele. Little r is wrinkled, that's the recessive. So in a dihybrid cross, you're taking all the different ways these alleles can combine. So in this first example, We have little r, little r. So we have big R, and then little r. And then we also have big Y, big Y. Oh, this is a little different. So I'm gonna scratch this out. We have big R with big Y, and little r, little y. So one offspring, the top parent can only donate a big R and only donate a big Y. And the bottom one can only donate little r and little y. So that's why on the side we have big R and big Y, because that's all that can be produced, big letters. And that's why on the top we get little letters, because that's all that can be produced. We're taking one allele from each gene and combining it together. So in this case, we always have the dominant trait. They're all yellow, all round. And this one, it gets a little more tricky. We're going to do with the top one, since the bottom was the same. We have big R, and big Y, little y. Remember, we can take a big R and a little r away from here. But we can also take the big Y and the little y. So one big R can be paired with one big Y, taking that. But since you're separating the Y's, the big R can also be paired with a little r, little y.